The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 37 of the eLife Podcast, which is produced by The Naked Scientists. I'm Chris Smith. This month, how human handedness happens, new clues to combating toxoplasmosis, and the consequences for sons of having a father hooked on nicotine. But first, damage and disease affecting the brain and the spinal cord are devastating. A person never recovers their lost function because the central nervous system has a very limited ability to repair and regenerate its tissues. So instead, scientists are turning to technology to create more effective assistive devices for patients with neurological disabilities, and they're getting better all the time. From Stanford University, Jamie Henderson.
So what we were trying to do with this paper is to demonstrate the ability to type using brain signals and, in fact, create a system that allows people to move a cursor on a computer screen and to type at anywhere between approximately 4 and approximately 8 words per minute, which is a factor of between 2 and 4 faster than what's been demonstrated before.
And how would that compare with, say, me on my smartphone if I'm tapping out a text message?
Well, I would say the average person probably performs somewhere in the 12 to 19 words per minute range. So we're beginning to approach that range, but we're still probably about half of what you can type on a cell phone.
But critically, you are doing this without a person needing to move a muscle.
That's correct. This is all done through reading out brain signals and translating them into movements of a cursor on a computer screen.
Well, before you tell us why the competition are doing less well than you are, how does it work?
We begin by implanting a tiny sensor on the surface of the brain. The sensor is 4 by 4 millimeters. It's about the size of a baby aspirin, with 100 probes that penetrate just into the outer layers of the brain. These pick up signals from the neurons. When those signals are read out, there's an amplifier that mounts on the top of a device called a pedestal, which attaches to our research participant's skull and protrudes through the skin. We attach a cable to the top of this. We run those signals out, amplify them, digitize them, and put them into a computer where they can be decoded. And we can then use our computer algorithms to understand what the brain is trying to do, what sort of movement the person is trying to make, decode that, and display it on a computer screen.
How long does it take after you put this into the person's brain for them to learn to think along the right lines to achieve meaningful control?
We found that our research participants are able to get control of the cursor fairly rapidly. It's pretty intuitive. People usually pick this up within several days and become very facile with it in several weeks. And the reason for this is that we are basing our decoders on tuning of the brain cells. So we ask our participants to imagine or attempt a movement of their opposite arm. We then pick up the signals from those brain cells, which are tuned to the particular direction of movement. So if I imagine, for example, moving my arm up and to the left, certain of those brain cells will increase their firing rate or will be more active with that movement, whereas certain other brain cells will decrease their activity. Each probe of the array that we've implanted can pick up signals from one or several brain cells. And by decoding the tuning of each of those brain cells, we can then predict where the person is attempting to move and use that to move a cursor.
Now, people have been eavesdropping on the brain's motor circuits and using them to drive devices in this way for a really long time. I mean, this is not brand new, the approach you're taking, is it? So what is the novelty here?
Well, there are actually several innovations that we use to achieve this level of performance. One is real-time system design and real-time programming. This allows us to very rapidly feed back cursor position to our participants. So the cursor position is actually updated every millisecond. It's faster than even 120 hertz display can display it. So this very rapid feedback allows participants to rapidly acquire where the cursor and the target is and to better control it. We also use digital filtering to clean up the signal. So it's really these innovations all together that allow us to achieve this level of performance.
What about the long-term stability of the interface into the brain tissue, though? Because For many years, people have struggled to do this with these chronic implantable devices. They find the signal degrades over time.
Well, unfortunately, that is still a problem. We do see signal degradation over time, although one of the participants in the study had the implant for approximately 3 years at the time that we performed these research sessions, and her performance was among the best in the study.
Now, if you ask the people who used them, what was the approval rating? In other words, how did they respond? And also, given that you're now doing this in real time, what can you do to improve this further? Because we're not quite up to a teenaged text rate yet, are we?
No, we're not quite there yet. Participants very much enjoyed using the system. One of them said that after he had been using our system for a period of time, he went back to his old head tracking system and found it somewhat cumbersome and clunky. There's obviously still a lot of work to do. We do want to be able to type faster, as you mentioned. It's possible that by reading out the planning phase of movement, in other words, when someone is beginning to plan to make a move, it's been shown in the laboratory that one can very rapidly decode an intended movement even before a movement can be generated. So hoping at some point to explore that concept further.
And we hope you succeed soon. Jamie Henderson there. Our realisation that epigenetics offers our offspring an opportunity to adapt on a timescale far faster than a genome message can change has given us new insights into the basis of a number of diseases. Take diabetes and hypertension, for example, which are more common in the offspring of parents with metabolic syndromes themselves. But are the epigenetic changes that underpin these altered disease risks a highly focused response to just one component of an adverse environment, or a more generalised genetic alarm signal whereby the body alters its resilience against a range of potential threats in response to just one stimulus? Oliver Rando thinks it's more likely the latter.
Over the last decade or so, it's become increasingly clear that the environment experienced by a father can affect the health and the behavior of his children. Many of the environments chosen include dietary regimens or stressors that affect many aspects of a father's health. In this study, we wish to understand whether or not fathers tell their children much more specific information about the world they saw. To do this, we chose to use nicotine, both because nicotine is a commonly used drug in human populations, and so this would be relevant to human biology, and also because nicotine has a very specific molecular function, which is that it binds to a receptor. And so we could ask whether or not children are specifically more or less responsive to nicotine and only nicotine, or whether or not children become generally drug resistant or generally anxious or something of this nature.
In other words, is there a very specific and tight programming for a certain stimulus and a certain outcome, or is there more like a molecular tripwire here, where a range of things can sound the alarm and it then puts in play a whole range of responses, and it wouldn't matter what trip the tripwire, you'd see the same range of responses?
That's an excellent way to put it. We do this in inbred mice. One of the reasons to use inbred mice is so that all the mice have the same genomes. We treat pairs of male mice. Half of them are exposed to nicotine. They're drinking water, actually. The other half consume a control solution. We then take those mice and we allow them to mate with females. Then we take the male mice away so that they don't directly interact with the children. And then we analyze a variety of behaviors and phenotypes in the children.
And what trends emerge?
Well, so quite a few things don't change in the children. They don't become grossly more or less sensitive to nicotine. However, the children of nicotine exposed fathers become more resistant to toxic doses of nicotine.
This is like a sort of defense priming. If your parent has been exposed to X, then you are now primed to encounter things like X and defend yourself against them.
That's correct. And that's certainly how it seemed at first. The next part of the study was to ask whether or not the children are primed for X or whether or not the children are primed for a broader range of things. To do this, we also exposed the children to cocaine. And much to our surprise, even the children of fathers exposed to nicotine, those children became more resistant to cocaine as well. So that now tells us that even though fathers were exposed to one drug, the children become more broadly resistant to toxic levels of drugs.
We're using this term children, and that could mean male and female, couldn't it? Was there a difference between the male and female offspring? Did they both respond in this way, or was it just one of the two?
We only see the sons exhibiting changes in resistance.
Interesting. So how do you think that's being passed on then?
Well, we have no idea. We haven't done any molecular work on the sperm, which is, of course, the likeliest place for this information to be. In other types of paternal effect system, so in other experiments where we treat males with different environments and then look at the children, we are very keen on the idea that small RNAs are the carriers of the information about the environment. But in this particular study, we did not look in the sperm at all.
Well, let's explore, why do you think this effect exists at all?
Well, it certainly can be construed as being adaptive. By adaptive, I mean useful for the kids. If fathers are exposed to nicotine, their children become more resistant to this toxin. So that's certainly something that you can imagine being evolutionarily advantageous. Why the children become more resistant to multiple toxins, I'm less confident in speculating about. But at least in the case of when the fathers are in an environment where there's lots of nicotine about, the children will be better suited for that environment.
Obviously, these are rodent studies, and we've got to be cautious about translating them to humans. But what would be the implication then if you have a child growing up or developing in an environment where the father's being exposed to various chemicals? What might be the implications for that child's own use of nicotine or exposure to nicotine subsequently?
Well, in the mouse model, we did look at self administration, which is a model for addictive behavior. We saw no difference in the self administration of nicotine in these offspring. But that cannot be extrapolated to humans particularly well because mice are a notoriously bad model for nicotine addiction because the half life of nicotine is much shorter in mice. So while we have no evidence that it would affect predisposition towards smoking, studies in humans will need to be done to really make sure that that's true.
One wonders what trouble we may be brewing up for our children. That was Oliver Rando. He's at the University of Massachusetts. You're listening to the eLife podcast with me, Chris Smith. And still to come, evidence that your handedness comes from your spinal cord rather than your brain. Before that, though, at first glance, a parasite caught from a cat litter tray doesn't sound like it should have very much in common with malaria. But malaria is a very close relative of toxoplasma, and the discovery by Glasgow's Marcus Meissner that when toxo grows inside our cells, it assembles a communications network that synchronises the growth of the parasites could reveal new ways to combat both bugs.
Toxoplasma is a single-celled organism. It's a parasite that can only live inside other cells. It is commonly infecting cats, but it also is carried by other warm-blooded animals, including us humans. And amazingly, up to 1/3 of the UK population is actually chronically infected with this parasite, meaning if you get once infected, you will carry this parasite within yourself for the rest of your life, mainly as tissue cysts in your brain, but also in your muscle tissue.
And how do most people pick it up?
There are two major routes for the infection. One is directly from the cat. Eggs are shed with the feces of the cats, up to 2 million a day. And the other route is via eating undercooked meat.
And once a person picks it up, what happens then?
Then the parasite starts with the acute infection, which is usually harmless for most people. It's like a common cold or flu-like symptoms. However, then the parasite differentiates into persistent forms, and these form tissue cysts in the brain and in the muscle. And so the medical importance is for immunocompromised people where the immune system breaks down. There the tissue cysts reactivate and it can cause encephalitis and brain damage. And the other problem is if a pregnant person gets infected for the first time, the parasite can cross the placenta and infect the embryo, and that can cause heavy organ damage.
And what have you been trying to find out about it?
It's obviously already important to study it for its own right as a human pathogen, but it's also a very good model system for other parasites of the same family, the apicomplexans, which includes malaria. And what we wanted to find out is how the parasite actually invades the host cell, because in order to survive, the parasite has to invade a host cell. In case of malaria, that's our red blood cells, and also how the parasite replicates inside the host cell. So we focused our attention on a protein called actin, which is a scaffolding protein found in many different cells. However, in these parasites, no one knew if this protein can actually form a scaffold. And we found a new way of imaging this protein inside the cells and found a really amazing network that is formed between individual parasites in the cell, which the parasites use to communicate with each other, to exchange material, and to make sure that they have a synchronized cell cycle, meaning that all parasites replicate at the same time.
So we've got this situation where this parasite gets into a cell, whether it's malaria or, in this case, toxo, and once it's in the cell and it starts to grow or replicate, it's orchestrating this scaffolding, which is connecting the replicating or growing progeny together, both chemically and physically, and that seems to play a big role in how they grow and develop.
Yes, that is correct. So we find, for example, that if we disrupt this scaffold, the parasites are sort of crippled, meaning they cannot really finish their replication cycle. And you end up with parasites that cannot leave the host cell anymore. So they are trapped inside the host, and that is, of course, a way to interfere with the further transmission of the parasite.
Just before we come on to what the implications of that might be, what was the imaging technique that you used to enable you to see what these parasites were doing inside cells like this?
The trick was really that an antibody can be expressed in the parasite that binds to the scaffolding protein only when it makes a scaffold. And this antibody gives a fluorescent signal, and then using super resolution microscopy, we could make a 3D model of the scaffold, and using time-lapse imaging, meaning we made movies of the behavior of the parasite, we were able to identify material that is transmitted or transferred in between the parasites.
Do you think it's something as simple that if all of the parasites replicate or grow in a cell at the same time, then they're going to be able to share the resources that they're making at the same time? And that's a very efficient way, because you've got a big job lot of what you need all in one place at one time. You share it all out and you can grow much more effectively.
That is actually one of our hypotheses. At the moment, we were able to detect and describe the network, but we are not really 100% sure yet what is the exact function of the network. So we don't know the mechanisms yet.
Marcus Meissner from the University of Glasgow. One of the striking things about humans is that, in fact, 90% of us, when handed a pen or a tool, will use it with our right hands. And this is not a modern phenomenon either. Cave paintings suggest that our ancestors, even tens of thousands of years ago, were also very much more inclined to use their right hands for specialist tasks. So how does this happen?
My name is Sebastian Ocklenburg from Ruhr University Bochum in Germany. The question I'm really interested in is, how does handedness develop? We know that in the population about 90% of individuals are right-handed, but it's still completely unclear what determines whether somebody is left-handed or right-handed. The defining theory for the last couple of decades has been that handedness is determined by a single gene. But we know from genetic research in the 2000s that this is very unlikely. So in the 1990s, a researcher named Peter Hepper had the theory that handedness might not be determined by the cortex, but by the spinal cord, because it was found that embryos already show a right-handed preference for motor movements during a developmental phase in which the spinal cord is not connected to the motor cortex.
What did you do then to try to see how the spinal cord might be arriving at that situation of that right-hand-sided dominance in order to be future right-handers?
One of the major theories about development of handedness is that gene expression asymmetries might play a role here. So this is what we looked at. We tried to find genes that were expressed asymmetrically in the spinal cord. If there are expression asymmetries, that might be an explanation for behavioral asymmetries later in life.
How did you study that?
So we looked at tissue samples from the spinal cord from human fetuses obtained from a gynecological clinic. Those were from week 8, 10, and 12. So we looked at genes and how they were expressed on the left and right spinal cord. And we also performed epigenetic analysis. So we looked at asymmetries in gene methylation and expression of microRNAs.
Right, so what you're saying is that the genetic code may well be the same in an individual who's right and left-handed, but you can actually change how turned on or turned off certain genes are, and also the epigenetic markers which are added to the genes could be different on the right and the left, and that could account for the handedness, even though the genetic messages are the same.
Exactly. So we think this might be one of the reasons why previous researchers have found so few results when looking at genetic variation. Exactly that such processes might switch on or switch off or modify gene expression, which then leads to variation in key structures in the central nervous system that modify the behavioral phenotype, and you cannot see that from just looking at the genetic variation.
And was that clear? Could you see that there was a difference in the way the genes were being turned on and how much they were turned on the right versus the left?
So yeah, we found that these processes are very much dependent on the time point where we look at it. So if you look at the time point where the fetuses show this right-sided preference for this first time, you see a huge asymmetry in gene expression and also in these epigenetics processes, which gets less and less the farther it proceeds in development.
Now, famously, in his book Right Hand, Left Hand, Chris McManus, who did literally write the book on left-handedness since about the 1980s, now he highlights in that book that asymmetry begets asymmetry. In other words, just because you have found an asymmetry in the gene expression in the spinal cord, well, something must produce that asymmetry in the first place. So what do you think, or how do you account for the fact that you've got this difference in expression of gene patterns on one side of the cord versus the other?
So that's probably the biggest open questions I want to address in my research in the future. Which environmental factors actually trigger these epigenetic processes? I can't really say I have a good clue at that right now. Some people, for example, suggest stress might play a role. Some other people suggested that intrauterine hormonal environment, for example, testosterone level. But it's really something that we will have to look up in follow-up experiments. I cannot really answer that right now, I'm sorry.
So there is still a mystery to solve. Sebastian Ocklenburg there. Each month, we try to devote at least one of our interviews to considering some of the wider impacts of scientific practice and scientific life. Recently, we've looked at the impacts of the positive results bias, how women fare in science, and also whether science is losing its best young talent owing to funding shortfalls. This month, with the help of assistant features editors Sarah Shales and Stuart King, we're considering eLife itself and the role of its research digests. And if you're not sure what a digest is, Sarah can explain.
eLife digests are plain language summaries of research articles, and we've been including them in research articles since the journal launched in 2012. They always start with some background information about the area of the research, and they'll set up the kind of research question that the authors of the research article were trying to address with their research. And then they'll talk about the main findings of that research, and then they'll go on at the end to talk about the next steps and kind of the relevance to the research.
How long are they?
The average digest is 347 words, and I know that because we recently looked back, but the range would be about between 200 and 400 words.
So that's on par with a news piece, isn't it, that you'd see in a newspaper?
Yeah, so they're short, complete articles on their own basis. So we do take a number of the articles and we share them on a separate site called Medium. We do that to reach a different audience who aren't already reading the research articles.
What's your motivation for doing this, Sarah?
Our main motivation is that we want to make findings of research papers more accessible to a broader range of people. That includes other scientists from other fields of research. I'm a plant scientist by training, and when I try and read a neuroscience paper, I find it very hard. There's all sorts of jargon that I don't understand. By using much more simple language and active language, so talking in terms of processes happening as opposed to kind of using lots of nouns in the sentences, really helps people who aren't specialists to get hold of that information and use that. We try and aim digests to be accessible to people who are sort of towards the end of their school years and adults who are interested and motivated in science.
Why should the journal do this? Because you could argue it's for scientists, and scientists know a lot of these words anyway.
One of the major motivations is because we're an open access journal, all of our research articles are freely available to read by anyone. So adding the digests in is part of that, in that the audience could be anyone and they can access and read and understand the content. If you want your research to have an impact in the real world, you need to reach beyond the academic community. A digest, although it's a small summary, is a small step towards doing that. One of the other motivations for us doing this is that we get the authors of the research article involved in the writing.
But you haven't always done it like that, though, have you?
I mean, we've always produced digests, but when we started, digests were written by us and by freelance writers who were reading the manuscripts, working out what the important messages were from the paper from that. A couple of years ago, we started asking the authors of the research papers to answer some questions for us in more plain language, and that's really helped us. It highlights what authors think is the main message of their research, and that's been great because we get different voices coming through in the digests because everyone thinks about research in different ways. But it's also been great in that when we send draft digests to the authors to check, they've made fewer changes because actually what we're sending them is something much more similar to what they've seen before, and I think we land up with something that the authors are overall more happy with at the end.
And more importantly, what do the audience think? Because the rule of any medium is give the audience what they want.
That is something that we've looked into relatively recently. At the end of last year, we conducted a survey of current digest readers, and we had over 300 responses. Overwhelmingly, I think the response was fairly positive. Most of the audience are scientists, but we are also reaching non-scientists as well, and that's an audience that we're probably looking to engage more with. But on the whole, both scientists and non-scientists alike find the digest that as we currently write them useful, good structure, good language. So we were very pleased with the results of that survey, actually.
What about other journals? Because lots of journals do go down this same path. So they're offering a similar sort of thing, aren't they? A similar synthesis of what you need to take away from this paper.
Yes, so while we're working on this series, we found about 50 journals that actually produce plain language summaries, and I wouldn't be surprised if that's not all of them. One of the things that strikes me about them is there's such a variety out there. They're written in different styles, they're different lengths, they're aimed at different people. So some organisations, for example, the journal Autism, are particularly targeting their summaries at patients and their families.
Do they think this is useful?
Definitely. One of the articles that specifically looked at this exact issue and patient involvement and how plain language summaries can help that. So HD Buzz is an organisation that covers the latest research in Huntington's disease, and they've found that for those families that are affected, finding out about the latest research provides them with real hope to deal with these conditions happening in their families. I think it's a strong motivator for the researchers involved.
But isn't that what a newspaper's for?
Yes and no. So another organisation that's looked into this is Inspire, which is a collection of disease-related social networks. And they've found that patients and their caregivers look into a range of different sources. Over half of them are looking at journal articles. But the slight issue with journal articles is it's not written for them. It's written for other scientists. So it often doesn't quite give them the context of what it means for them and how close it is potentially to a treatment. Newspaper articles, on the other hand, might overemphasize that. They'll focus too much on that and get the headlines, compound X cures cancer. That's often grossly oversimplified. Another organisation looking to tackle this is Dr. Media, based in the Netherlands. And it's been inspired by the exact conversations, patients coming to their doctors asking for guidance on what a newspaper story means for them. And they work with clinicians to produce this content because in one-to-one consultations, you're only working with one patient, but by making it accessible online, they can reach a much wider audience.
Were you surprised by what you found?
Yeah, I think we were pleasantly surprised. Our biggest fear when we went into the survey was that we'd find that no non-scientists were reading eLife Digest.
And then you'd be out of a job.
Yeah, it would feel like the aim of them was being missed, really. So the fact that there are non-scientists interested and motivated enough to find them was really nice and reassuring, I think.
Hear, hear. That was Sarah Shales and Stuart King. And Sarah and Stuart's Inside Guide to eLife Digest, their analysis of plain language research summaries, is online now at elifesciences.org, which you can also go to find all of the papers that we've discussed this month. You've been listening to the eLife podcast, previous editions of which are also on eLife's website, or you can subscribe on iTunes. This is a Naked Scientist production. I'm Dr. Chris Smith, and you can find out more about what we do on our website at nakedscientist.com. I'll be back with another look inside eLife next month. In the meantime, thanks for listening, and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.